the first session of the day really focused on this high-level infrastructure, programmatic initiatives that support a lot of the effort. All of that obviously pairs with data modalities, data generation, and the next set of talks really intersect across, at least in my mind, two domains. One is what you heard from James, Coulomb, and others on the opportunity landscape around cross-disease discovery, germline predisposition, germline-based analysis in this context. But the second will also be multimodal data. The release that just happened within CBTN includes, by and large, whole genome sequencing and RNA-seq for this largest brain tumor atlas cohort, paired with imaging, some microRNA-seq data. The separate EXO-1 initiative is focusing on proteomics and phosphoproteomics as a context, but you'll hear from many of the teams about to speak about this intersection between germline context, functional genomics, additional assays, and the multimodal context that bridges into the, in my mind, translational setting. Our first speaker is going to be Jeremy Shaw. This is my first time meeting Jeremy, but I know his postdoctoral mentor extremely well, Philip Lupo. And so he's partly filling in for Lupo, but I think it's to Philip's testament that what Jeremy's doing at Baylor and what he'll be talking about. The third thread here will be additional programmatic intersections between not only Kids First, but also with NCI-based initiatives, and you'll hear about that from a couple of speakers. So I'd like to welcome Jeremy from Baylor.
All right, well, thank you, Adam, for the introduction, and thank you to the organizers for including our work in this session today. This is my first time attending the summit, so very excited to be here. So I'm going to speak a lot about birth defects, cancer risk in children with birth defects. I might use the terms birth defects, congenital anomalies, or congenital malformations synonymously throughout my talk. And I'm going to use them to refer to any of a set of structural or functional abnormalities that are present at the time of delivery. As has been mentioned, there are a wide array of different specific birth defects that have been characterized and that can occur, but if you consider them all collectively, they affect between 3 and 5% of U.S. live births. So these are not especially rare conditions considered as a group. They're also notable for being the leading cause of infant mortality. About 1 in 5 deaths during infancy are attributable to congenital anomalies. And there are more than $23 billion in direct hospital expenditures annually for the treatment of affected people. Our interest in the intersection between birth defects and cancer goes back about 10 years now, kind of parallels the timeline of the Kids First initiative, more or less. And at that time, there had been some case series and some clinical studies that had noted an excess of congenital anomalies in children with cancer. That had caught the attention of clinicians and obviously of the research community. And we started to see some population-based data come out that suggested probably 2 to 3-fold increased risk of cancer in children with birth defects. There are a number of potential mechanisms for that James already discussed. Certainly there are probably common genetic causes in some instances. There may also be common environmental causes, factors like maternal diabetes or folate insufficiency that could contribute to these co-occurrences. And potentially also iatrogenic exposures, particularly in children with congenital heart disease, things like ionizing radiation exposure from CT scans and cardiac catheterizations. But with respect to brain tumors in particular, I think the picture was a bit more mixed. Some studies had suggested increased risks of cancer in children, of brain tumors in children with birth defects, particularly CNS anomalies, where there's about a 6-fold increase in the hazard in one study. However, other population-based assessments had found no association. So, that left some important and unresolved questions. There was a lack of data, I would say, on specific birth defects and specific tumors. The interpretation of the data that we did have was complicated by the inclusion of both children with chromosomal or genetic syndromes, as well as apparently non-chromosomal structural birth defects. And I think it became apparent that large studies with really robust birth defects phenotyping were going to be needed to address those questions. So in 2017, I started a postdoctoral fellowship at Baylor, and I was working in Philip Lupo's lab. I'm guessing many people in the audience know Philip. He and his collaborators had begun work on what they were calling the genetic overlap between anomalies and cancer in kids, or GO-BACK registry linkage study. This was a project that involved 4 U.S. states. We were linking data between population-based birth defects registries and population-based cancer registries. and vital records, essentially forming a cohort of more than 10 million US live births for whom we had information on birth defects and cancer diagnoses. And that was a big lift. I spent a lot of time during my postdoc cleaning and harmonizing and then analyzing those data, but I think it was worth it in the end, because with the benefit of the very large data set that we assembled, we were able to look at birth defect cancer associations in a much more granular way than other studies had been able to. So this heat map shows you on the y-axis sort of indicator variables for whether or not a child had an anomaly in any of a few different organ systems. And then on the x-axis, we're looking at some of the more common pediatric cancers. There's a little bit of a formatting issue here with these boxes that are meant to highlight results, but you can see CNS tumors. I think the fifth column from your left there were certainly associated with CNS anomalies, as we might expect in this cohort, but also with anomalies in a variety of other organ systems as well. In fact, everyone that we investigated, with the exception of orofacial clefts. And that was generally true for astrocytoma, for medulloblastoma, associations with CNS anomalies, but also non-syndromic anomalies in a variety of other organ systems. For context, those associations were not quite as strong as for some other tumor types that we looked at, like hepatoblastoma and germ cell tumors, but they were certainly more striking than, say, for acute lymphoblastic leukemia, which is really only associated with trisomy 21 or Down syndrome in our assessment. We went a step further, and we looked at a number of associations between specific anomalies and specific cancers. Again, I highlight a few results pertaining to congenital heart disease and various embryonal tumors, because I think these are some of the most interesting findings to emerge from that initial analysis. But we did also see associations with both chromosomal or genetic syndromes, as well as non-syndromic anomalies and CNS tumors, confirming, for instance, and generating a population-based risk estimate for astrocytoma in children with neurofibromatosis. Also demonstrating that, some of these associations are probably not things that we would have hypothesized out of the gate. For instance, a doubling of the risk of astrocytoma in children with craniosynostosis, and a six-fold increase in the risk of medulloblastoma in children with pyloric stenosis. We did also look at ependymoma, which was sort of more specifically associated with CNS anomalies, but those associations were quite strong, quite striking in this study. Concurrent with this, we had developed complementary IRB protocols in each of these states that would allow us to recontact families of individuals where the proband had both a congenital anomaly and a cancer diagnosis. And we did this in order to develop a family-based cohort for sequencing experiments, with the intention of looking both at single nucleotide variants and small indels, as well as larger structural variants that might contribute to the co-occurrence of these phenotypes. We called that the Go Back Family Cohort, and we were successful in enrolling 47 families into this cohort. When we generated those sequencing data, we noticed immediately that 11 or 24% of the probands had a pathogenic or likely pathogenic variant that we felt was related to the phenotype, which is higher than we would see probably in an unselected pediatric cancer population. Broadly, the variants that we identified fell into a couple of categories. There are some individuals with variants that were known to be associated with cancer predisposition in genes like TP53, others that were known to be associated with congenital anomalies, and a couple like WT1, which have been linked to both. But I'm going to talk more today about two variants that we identified in genes called USP9X and CAT6B. So both of these had previously been linked to congenital anomalies, but in this cohort, we identified a female proband with a de novo 5kb heterozygous deletion in an X chromosome gene called USP9X. That was work that was led by Samia Sasudia, who was then a postdoctoral fellow in Sharon Plon's lab at Baylor. So you can see this deletion in the affected female, absent from other unaffected family members. That particular child had a multiple birth defect phenotype that included coloboma, heart defects, choanal atresia, and other features, and they had a diagnosis of precursor B-cell acute lymphoblastic leukemia. That was significant in our opinion because we had observed an association between choanal atresia and acute leukemia in our registry analysis with a hazard ratio of just over 9 in affected children. And we had also shown that children with multiple major anomalies, but no diagnosed syndrome were at the highest risk of cancer. So this was consistent with what we had seen in our registry-based analysis. There's also a gene that we felt was very biologically plausible. So this is an X chromosome gene known to escape X chromosome inactivation, encodes a highly conserved deubiquitinase enzyme that participates in several relevant developmental pathways like EGF, Wnt, and TGF-beta signaling. So by reviewing the literature, Saumya and the team were able to identify additional females with loss-of-function variants in this gene. You can see individual patients on the x-axis, including the patient in GOBAC, and on the y-axis, you can see various phenotypes that have been reported in these individuals. You can also see by looking at the rows near the bottom that there were 3 additional cases with cancer, 2 with BALL and 1 with osteosarcoma, and that there were different classes of variants occurring across the gene body as we would expect. Finally, we realized that it had been reported previously that USP9X is actually one of the most frequently somatically mutated genes in the context of pediatric cancer, with those mutations occurring in the context either of B or T ALL. So we concluded that USP9X is a novel ALL susceptibility gene with a charge-like phenotype. A similar example is CAT6B. So this has been implicated in genitopatellar syndrome, clinical variant of Odo syndrome. Phenotypes associated with those conditions include musculoskeletal defects, congenital heart disease. But when we identified a CAT6B variant in a case with neuroblastoma, we began to suspect that neuroblastoma risk might be an additional feature. We had found some associations with defects that are commonly observed in the context of genitopatellar syndrome in our association analyses. But we wanted to dig into this in more detail. So we used data from the Kids First Initiative, 400 neuroblastoma cases, about 900 unaffected individuals. We conducted burden of rare variance analysis using the VAST algorithm, which revealed evidence of significant enrichment of predicted deleterious CAT6B variants in neuroblastoma cases relative to unaffected controls. And then we performed a series of logistic regression analyses where we looked only at variants that met the criteria that you see here on the left and saw consistently elevated odds ratios of neuroblastoma, leading us to believe that CAT6B is also a neuroblastoma susceptibility gene. So, thus far, I think this work has been productive. It's identified numerous birth defect cancer associations that could be the targets of future investigations, contributed to the identification of at least 2 cancer predisposition genes, and demonstrated that the analysis of this sort of population-based registry data can inform genomic studies of pediatric cancer. At the same time, it also highlights the utility and the importance of the Gabriella Miller Kids First sequencing data to help elucidate the mechanisms underlying the sorts of associations we're seeing in the population. So I want to take just a couple of minutes to talk about future directions, both in the context of sequencing studies and our association analyses. We were very fortunate and very grateful to receive some support for additional sequencing through the XO1 mechanism. We're leveraging the Children's Oncology Group Project Every Child protocol for this work with the sort of 3 goals. Firstly, to identify the prevalence of known cancer predisposition variants in children with congenital anomalies in cancer, to characterize novel ones, and then also to evaluate the somatic landscape in these children. COG is probably familiar to many in the audience, but if it's not, this is a large pediatric cancer clinical trials network, has more than 200 sites in the US, many more internationally, and about 90% of children with a newly diagnosed cancer in the US are treated at a COG member site. So we were able to identify 500 individuals with cancer and congenital anomalies through COG's Project Every Child protocol, which has really been a boon to epidemiologic and etiologic research. That included 73 cases with brain tumors who had a variety of congenital anomalies. We have generated genome sequencing data at 30x coverage, exome data at 100x coverage, and furthermore subjected 120 of these samples to transcriptomic profiling. Simultaneously, we're continuing to develop the GOBAC registry linkage cohort. We've expanded it to 5 additional states, updated linkages in states where we had existing protocols. And I'm thrilled to say that we now have more than 22 million children in the cohort. So we're going to use that to evaluate rare tumors, look at multiple congenital anomaly patterns, and do a variety of deep phenotyping experiments that I look forward to sharing with you in the future. Project Every Child includes some limited information on birth defects diagnoses, but we are working with Adam and others to try to expand on that and get some deep phenotyping data that we can include with the sequencing data to try to increase the utility of that data set when it becomes available to the community next year. So quick summary, I think the registry analyses that we performed demonstrate that birth defects are associated with increased cancer risk, including CBTs. We see the highest relative risks in children with multiple major anomalies. And if we consider all birth defects collectively, the population attributable risk might be on the order of 7 to 10%, which I think is pretty extraordinary for a single exposure in the context of pediatric cancer. Genomic studies have also been fruitful. They're identifying genes with pleiotropic associations involving both birth defects and cancer, and seem to have a particularly high diagnostic yield in individuals with both congenital anomalies and cancer. So, of course, I want to thank everyone who contributed to this work over the years. Very long list. This is just some of the folks who assisted with that project. I want to thank all of the patients and families who made the research possible. Thank our funding agencies, and I'll turn it back over to Adam.
Hopefully you noticed, right, that the initial study had 73 patients, right? And I think this is one of the opportunities that CBTN really sort of positions is to really intersect between, COG, CBTN, and other initiatives. You'll hear a lot more of that from Sarah when she talks later. But the other part, as I was sitting there, remember, we just released a ton of data on Kids First. So I was searching for your favorite genes in the new CBTN cohort. And I think there are more than 20 kids in CBTN that have CAT score over 20, right, mutations. So you can do this in real time as you're watching essentially presentations.